Good afternoon, everyone. Welcome. I'm Jack Van Horn from the University of Virginia, and it is my distinct pleasure to welcome you to this edition of our 2024-2025 Foundations of Biomedical Data Science Seminar Series. Our unique lecture series is made possible through the generous support of the Integrated Translational Health Institute of Virginia, the University of Virginia's College of Arts and Sciences, the School of Data Science, and through a grant from the National Institutes of General Medical Sciences. Since its founding in 2016, the Foundations of Biomedical Data Science Seminar Series has looked at the forefront of exploring transformative impact of data science methodologies, artificial intelligence, deep learning, and statistical modeling across a diverse set of themes in biomedical and health sciences. Our selected theme for this year is focused on quantitative approaches in spatial multi-omics for guiding personalized medicine, where every Friday we enjoy presentations from a wide range of leading thinkers about the issues, the promise, the opportunities, and the hurdles associated with advanced genomic, transcriptomic, metabolomic, and other omic-related methods, along with a wide variety of data analytic challenges presented in data science, deep learning, AI, and spatial modeling. We are particularly pleased to announce that our 2024-2025 application cycle is open, and we hope that you or any of your junior faculty colleagues working in various areas of omics-related biology, as well as clinical medicine, will be interested in applying. Aligning with this year's theme, we are eager to receive applications from folks working in multi-omic data analytics, computational biology, machine and deep learning, spatial statistics, network analytics, graph metrics, as well as software design and user interfaces. Participants selected for our special Biomedical Data Science Innovation Lab program will utilize the content from lectures such as the one we're about to hear for our culminating in-person manuscript and grant development workshop to be held at the Hutton Hotel in downtown Nashville, Tennessee in June of 2025. We're excited by this year's program where these junior faculty folks will get together, form unique teams, and propose novel projects looking at spatial multi-omics and the data science that it utilizes. For today's lecture, I am delighted to welcome our speaker, Professor Jason Stein from the University of North Carolina in Chapel Hill. Jason is an associate professor in the Department of Genetics and the UNC Neuroscience Center. His research focuses on understanding how genetic variation alters brain development, structure, and function, and he has long been involved with the Enigma Consortium and has conducted several invaluable examinations on the role of gene SNPs and brain structural differences in patients as well as controls, and his work has appeared in many leading periodicals such as Nature, Nature Neuroscience, and other major venues. In the spirit of our 2024-2025 Biomedical Data Science Seminar Series theme, Professor Stein's lecture today is entitled Genetic Effects on Microscale and Macroscale Brain Structure. Genetic differences between humans lead to variability in the structure and function of the brain, and through the collaboration of many different labs across the world, leading to analyses of over 30,000 participants, researchers have identified common genetic variants which affect human cortical and subcortical structures as measured through MRI. While these findings have provided insight into molecular pathways involved in determining human brain structure, the cellular basis of these brain structural differences is not known due to the limited resolution of MRI. To address this, Dr. Stein and his team conduct tissue clearing and lightsheet imaging of intact mouse brains harboring psychiatric disorder associations and human organoids to understand genetically mediated differences in brain cytoarchitecture. As always, we are streaming this lecture live and for recording via YouTube. If you're watching on YouTube, thank you for joining us. Also, our specially selected 2024-2025 Biomedical Data Science Innovation Lab participants, as well as alumni, are encouraged to submit any questions they have via the chat feature in their YouTube sessions. I will synthesize these questions and I'll ask them on your behalf during the last 10 minutes or so of the lecture. And with that, thank you so much, Jason. We really appreciate being here and we're really looking forward to hearing from you.
Great. Thank you so much, Jack, for the very nice introduction, and thank you for inviting me to speak here today. So I'm going to talk to you, as Jack said, about genetic effects on microscale and macroscale brain structure. So the spatial aspect of the spatial multi-omics that I'll talk about is the structure of the human cortex. So the human cortex is the outer covering of the brain. It's greatly expanded in humans relative to our closely related species. And specifically, the cortical surface area is greatly expanded in humans relative to other species. It's thought that human gained cognitive and social abilities are possible in large part because of the expansion of the neocortex. In addition, individuals with many neuropsychiatric disorders have altered cortical structure, implicating this structure in psychiatric disorders. So genetic variation clearly is one part of what has led to an overall increase in cortical size. So how do scientists generally identify the genes that are involved in cortical and brain size? So on a human genetics approach, classically, we study microcephaly or macrocephaly cases, individuals with very large or very small brain sizes. And those individuals are rare in a population, and we can sequence the genomes of those individuals and identify genetic variants that lead to risk for microcephaly or macrocephaly. So here's an example of one of those where a translocation breakpoint near a gene that's known to be critical in brain development is associated with microcephaly. Those type of mutations can also be inserted into mice, and we can study the effect of those mutations on brain structure. Here, observing a slightly smaller brain in knockouts of the gene EOMES. So this is classically how we study genetic variants that lead to differences in brain structure. But there are far more types of variants that exist in the genome. A set of those is called common genetic variants, and common genetic variants exist in all of us. And common genetic variants likely lead to inter-individual differences in the size of the cortex. So, are there in fact inter-individual differences in the size of the human brain and specifically the cortex? So here you can see the 2.5th to 97.5th percentile of differences in cortical surface area for males and females. This is basically equivalent to a 16-inch pizza versus a 20-inch pizza in terms of total overall brain size. So what determines the differences in brain structure across human populations? Is it genetic? And if so, which specific loci in the genome lead to differences in structure? This is what our study tries to ask. So in order to do these type of studies, finding genetic variation associated with macroscale brain structure, we need really large sample sizes of individuals who have both undergone MRI, where we can measure brain structure, and genome-wide genotyping, where we can identify genetic variants that exist in a population and determine if certain alleles are associated with inter-individual differences in the size of the brain. Here in my toy example, the A allele is associated with increased brain size, whereas the C allele is associated with decreased brain size. So the first studies I'll talk to you about are going to be how we have identified genetic variation associated with macroscale brain structure. And then in subsequent studies, I'm going to tell you about how our lab has tried to identify how those genetic variants alter gene regulation, leading to differences in overall brain structure. So this will be the sort of levels of multiomics that we'll cover, at least initially here, which are genetics and gene regulation, including chromatin accessibility, which is a proxy measure for transcription factor binding. And basically, it allows you to understand regulatory elements in the genome, as well as gene expression. Okay, so let's dive in. Our first study that I'm going to talk about measured cortical structure using MRI. In this type of study, we take pictures of the brain with MRI and large sample sizes of individuals. Those pictures can be processed so that we segment out regions of interest. In this case, the cortical ribbon, shown here colored. And the cortical ribbon can also be divided into different regions based on gyral and sulcal anatomy. So from each of the different cortical regions, we can quantify features such as surface area and thickness of the cortex, the distance between the gray matter, white matter boundary, and the pia. And this can be done, importantly, automatically and in huge sample sizes, in very large sample sizes. So when I was a graduate student, actually down the hall from Jack, we founded a consortium called the Enhancing Neuroimaging Genetics Through Meta-Analysis Consortium, the ENIGMA Consortium. We started this consortium back in 2009, and the goal was to gather together enough individuals that we would be able to detect the effects of common genetic variants on brain structure. So a recent study from our Enigma consortium conducted GWAS in around 34,000 individuals with replication in an additional 17,000 individuals across 60 cohorts across the world. So this was a huge contribution from multiple different labs across the world to gather together enough sample size that we were able to detect for the first time significant effects of common genetic variants on brain structure. So we did identify many genome-wide significant findings. We had about 370 loci with a standard genome-wide significance threshold and 230 loci with a more stringent threshold that corrected for all of the different regions that we tested. So here you can see an ideogram showing all of the different chromosomes in the genome, at least the autosomes. And each of the colors here represent different regions that were tested. And the symbol represents either a significance threshold or the phenotype that was tested. So here you can see that actually we discovered many different loci across the entire genome that are associated with interindividual differences in the size of the cortex. Oftentimes, those loci are associated with just one brain region. They're not associated with every brain region, although some were associated with multiple different brain regions, as shown here. So overall, we have now a map in the genome of where genetic variation leads to differences in the size of the brain. Okay, so one of the first questions that we want to ask about this is we know the function of a lot of the genes in the genome, so we can determine if those genetic variants that are associated with differences in size of different areas of the cortex, are those genetic variants clustering near genes that are involved in some known biological pathways? And it turns out that this is the case, and it turns out that genetic variants near genes involved in the Wnt signaling pathway were enriched in these brain structure GWAS. Now, this was really exciting because Wnt is known to be a patterning factor in the developing brain that leads to differences in proliferation, giving support to our approach. So next, we wanted to try to get some additional information to determine if there were specific tissue types, developmental time periods, or cell types that contribute to differences in human brain structure. So we pursued this through what's called a partition heritability approach, which effectively asks if genetic association signal is enriched in defined regions of the genome. So those regions can be annotated based on gene regulatory functionality in specific tissues, time periods, or cell types. So in my cartoon example here, we can see that we've defined a region of the genome that are brain active regulatory elements or liver active regulatory elements. And by overlapping our GWAS onto these regulatory active elements, we can determine if there's a heritability enrichment within certain regions of the genome. So we first pursued this using what's called the epigenomics roadmap, which defined regulatory elements across basically the whole body. And what we observed is that the strongest enrichment for brain structure GWAS was found in regulatory elements of the brain. So this is somewhat expected, that regulatory elements, genetic variation within regulatory elements of the brain alters the structure of the brain. So kind of giving us a true positive result. So we then subset these elements to fetal specific or adult specific elements. And we also looked for progenitor elements or those elements that had specific functioning during Wnt signaling. And we again ran this partition heritability analysis, finding that especially for surface area, there was an enrichment in heritability explained during fetal development, especially in progenitor cells, and that regulatory elements in progenitor cells that were affected by Wnt signaling also show enrichment in cortical surface area heritability. So what does this imply? This implies that although we were measuring brain structure late in adulthood, that the genetic variants may be having their effect very early on during fetal development, during this period when neurogenesis is happening. So this kind of shows the development of the human brain from very early neural tube closure at 23 post-conception days, all the way up to here a newborn brain. And during this period of fetal development, where we see partitioned heritability enrichment, the process of neurogenesis is happening. So what could possibly explain these results? Well, we think that this result gives credence to what's called the radial unit hypothesis. The idea behind the radial unit hypothesis is that progenitor cells early in development make fate decisions. Those progenitor cells can decide to form other progenitor cells, or they can decide to form neurons. But neurons are postmitotic, they never divide again. So if progenitor cells make early fate decisions to form neurons, they may lead to a smaller cortical surface area. Whereas if progenitor cells make fate decisions to form other progenitor cells, which make fate decisions to form other progenitor cells, that expands the overall progenitor pool, which then leads to an enhancement of cortical surface area, an increase in cortical surface area. So what we think is that genetic variation within regulatory elements impacting progenitor-associated genes before birth impact adult cortical surface area. So kind of implying a mechanism for what leads to differences in overall human brain size. It's happening very early on in development. Okay, so interestingly, genetic association studies have not just been conducted for brain size, but been conducted for really a variety of many different traits. So we can use a tool called genetic correlation to determine if the same genetic variants that are associated with cortical surface area are also associated with traits like educational attainment, intelligence, things like neuroticism, major depression, etc. And what we found is that those genetic variants associated with increased intelligence in a population, as measured through IQ, are also associated with increased cortical surface area. In addition, those genetic variants associated with traits like neuroticism, depressive symptoms, major depressive disorder, etc., were associated with a decreased cortical surface area. So these genetic correlations imply that those genetics altering brain structure also alter the function of the brain as well, because they're associated with these other traits. Okay, so in this part of the talk, I hope I've convinced you that we have identified common genetic variation influencing human cortical surface area and thickness, that genetic variation influencing postnatal cortical surface area is enriched in active regulatory elements within the prenatal progenitor cells, which we think is human evidence for the radial unit hypothesis. Also, associations near Wnt signaling genes are involved in brain structure, implicating this known developmental pathway and shaping the human brain. And finally, that genetic variation affecting brain structure is partially shared with genetic variation affecting brain function. Okay, so overall, we've conducted this genome-wide association study. We've identified many loci of interest and have used some of that data to understand what processes are being regulated to impact brain structure. However, there's really a lot of difficulty in interpreting brain structure loci. So here's an example of a brain structure associated locus. Here we're focusing on the lingual gyrus here, and this is a map showing how much this particular SNP affects different regions of the brain. Most strongly, it's associated with changes in occipital cortex surface area. So if we zoom in, so this is kind of the spatial map in the brain, and we can also zoom in the genome to see a locus on chromosome 11 that is associated with differences in brain structure. So in that locus, each of these different dots represent a SNP, a single nucleotide polymorphism, a type of genetic variant that's polymorphic in the population. And those SNPs, you can see we have a lot of different dots that are associated, a lot of different SNPs that are associated with brain structure. Interestingly, none of these are changing the protein coding structure of any of the genes in the region. So most brain structure associated loci are in non-coding regions of the genome, and they're not changing the proteins. Also, because of something called linkage disequilibrium, which is sort of the correlation between nearby SNPs at a given locus, the causal variant within the locus are unknown. So genetic variants are inherited together during meiosis, so we can't actually separate which or if more than one of these are causal at this given locus. Also, the causal gene is unknown. Because these are non-coding genetic variation, we don't know if this genetic variant is impacting a close gene, a far away gene, or an even farther away gene. Genetic variants, although they exist in every cell in the body, may only have function in specific cell types or developmental contexts. So those are also unknown. The cell types or developmental contexts that these variants exert their function in are unknown. And finally, what the mechanism is that leads to the regional specific effects is also unknown. We see that genetic variant has more of a specific effect in one region than in the others. So how do we try to answer these questions and basically understand the molecular mechanism leading to changes in brain structure? So in order to understand this, one mechanism that we can use is called quantitative trait loci, QTL analyses. And the idea of these are very similar to the genome-wide association study. We obtain a population with inherent genetic variation, and then in that population, we measure differences in gene expression or chromatin accessibility as a proxy for regulatory activity. So by conducting associations between genetic variation and gene expression, we can get what are called expression quantitative trait loci, EQTLs. And by conducting associations between genetic variation and chromatin accessibility, we can do CAQTLs. If the same genetic variants associated with brain structure are also associated with chromatin accessibility or gene expression, we can begin to develop a molecular mechanism by which genetic variation leads to differences in enhancer activity, changes gene expression, and leads to downstream brain structure changes. There are several advantages to doing a QTL approach in that it models the endogenous genome function. So we're not sort of exogenously changing anything in the genome. However, there are some disadvantages to this approach as well. There is still confounding by linkage disequilibrium. It doesn't get around the linkage disequilibrium problem. And there's also background genetics that are not just the one SNP that we're studying, but there's many SNPs in the region or in the genome in general. So importantly, when conducting a QTL study, there is cell type and context specificity that one must worry about. So QTLs may only be observed in some cell types and not in other cell types, probably because transcription factors may only be expressed in certain cell types, leading to differences in gene expression only in cell type A in this case, but not in cell type B because the gene is not expressed and the transcription factor is not expressed. Similarly, there can also be context specificity. So a genetic variant may only exert an effect when stimulated by some compound, which leads to transcription factor translocation into the nucleus, and then leads to differences in expression across genotypes. So these context and cell type specificity of QTLs are something that my lab focuses on, and we try to model these using a human neural progenitor cell system. So we use a cell culture system to evaluate context and cell type specificity. We do this using primary human neural progenitor cells that were collected when I was a postdoc in the lab of Dan Geschwind at UCLA. We collected these from a lot of different donors, each of which we genotyped. Those donors, here we had for progenitors 85 different donors, and then we differentiated those neural progenitors into neurons. Not all of the donors survived, so we had around 66 donors. Importantly, cell cultures that we generate are heterogeneous, so we need a way to sort out the neurons from the other cells that exist in the dish. So we used a virus that specifically expresses GFP in neurons through a synapsin-1 promoter. So we had the progenitors and the sorted out neurons, and then we performed two different molecular assays, ATAC-seq, which measures chromatin accessibility, and RNA-seq, which measures gene expression. So ATAC-seq works by inserting a transposase that preferentially binds and inserts adapters into accessible regions of the chromatin, which we then sequence. Okay, so overall, we conducted a large-scale QTL study using both neural progenitors and neurons, allowing us to assess cell type specificity of chromatin accessibility and gene expression. So here's an example where I think we've been able to explain some of the molecular mechanisms of a cortical structure associated locus. So here we have a locus that's associated with the structure of the cortex, the cortical surface area. And you can see, like I showed before, this is a complicated locus. It's got many SNPs in LD. It doesn't change the protein coding structure of any nearby genes. So we have to layer on additional pieces of information from QTL studies to better understand this locus. So we layered on information from our progenitor EQTL datasets and our neuron EQTL datasets. And it turns out that this surface area associated locus co-localizes with the progenitor EQTL locus. So these are shared. What this implies is that those genetic variants that are associated with differences in surface area also are associated with differences in CENPW expression, specifically within progenitors, but not within neurons. We can explain even a little bit more by layering on CAQTL data. So for chromatin accessibility QTL data, we found that both in progenitors and neurons, that there was a CAQTL at the same locus. So this implies that this chromatin accessibility peak here, which turns out to be right at the promoter region of CANPW, is actually affected by this cortical structure associated SNP. And despite that there are many SNPs in LD, this particular SNP is the one to focus on because it's within the peak of interest. So it's likely that we've identified um, a cell type of interest, a chromatin accessibility peak, a gene of interest, and even a SNP of interest out of that complicated locus. Finally, it's interesting to note that this associated locus actually co-localizes with an educational attainment GWAS locus as well. And this is interesting because this genetic variant may not just be associated with differences in the size of the cortex, but also the function of the cortex as well. So overall, by layering on all of these different multi-omic methods, we can begin to suggest a cell type, a chromatin accessibility region, a gene, and structure that leads to differences in a trait that's correlated with cognitive ability. So to summarize this part of the talk, inherent genetic variation in a population of neural progenitors alters chromatin accessibility and gene expression during human neocortical differentiation. Cell type specific chromatin accessibility QTLs can lead to fine mapping and regulatory mechanisms of expression QTLs and GWAS loci. And overall genetic variation has impacts during neurogenesis that can influence adult brain structure phenotypes. And finally, integration of QTLs across multiple different biological levels allows for a mechanistic understanding of the impact of genetic variation on brain and behavior. Okay, so QTLs are one mechanism that we can explain those complicated GWAS loci. Another mechanism that we can use to try to explain those complicated GWAS loci is called a multiplexed parallel reporter assay. And this is some unpublished data that we're still working on, but I wanted to share here. So the idea of the multiplex parallel reporter assay is that instead of using inherent genetic variation in a system, we're going to induce genetic variation through a reporter construct. So in a multiplex parallel reporter assay, we take a sequence of the genome that's associated with macroscale brain structure. That sequence of the genome can be synthesized and then can be added to a random barcode. This construct can be then cloned into a reporter assay such that the element here drives expression of a reporter gene, and then we can measure the random barcode to determine how this element influences gene expression. If another element leads to higher levels of expression and therefore has higher regulatory activity, we would observe more counts of the barcode. Whereas if an element leads to lower expression of the random barcode, we would say it has less regulatory activity. So in this way, we can experimentally induce genetic variation and then see if that genetic variation leads to differences in gene regulatory activity. And because we're specifically selecting genetic variation associated with macroscale brain structure, these genetic variants will be related to the molecular mechanism leading to differences in brain structure. So there are several advantages of this multiplex parallel reporter assay, EMPRA. And one is that they model individual variant function, allowing the separation of background genetics and LD that's not possible with QTLs. However, there's also disadvantages to this approach. Here we're studying exogenous function in the absence of thingization, how DNA wraps around histones. So for these types of studies, we're studying a little piece of DNA without it integrating into the genome. Okay, so for the study that we're working on now, we actually have selected SNPs for inclusion in this EMPERA assay from the CAQTL study that I showed previously, as well as the Enigma GWAS study that I also showed previously. So for the CAQTL study, we selected SNPs and peaks that were where we found genetic variants associated with differences in chromatin accessibility in human neural progenitor cells. For the Enigma GWAS, we selected all SNPs that were in the LD block, so in the region that was associated, with a arbitrary threshold of p less than 1 times 10 to the negative fifth. So they have some nominal level of association. Importantly, there are differences in the number of SNPs that we studied across the different regions of the brain and across different traits. The number can vary because certain regions have very big LD blocks, and also certain brain regions we identified more loci that were associated with them. So there's a difference in, for example, the occipital cortex, we identified many more loci that were associated. Okay, so we put all of those elements together and we get 19,500 elements that we tested in total. So to give a little bit more about how we conducted the study, we took 150 base pair sequences. We took either of the two different alleles and attached them to random barcodes. The barcodes were 20 base pairs, and each element was attached to around, on average, 30 different barcodes. Those were then cloned into a plasmid and transfected into primary human neural progenitor cells with 14 replicates. After 24 hours, we then either added a Wnt stimulator, because the Wnt pathway we found to be relevant for differences in brain size, or vehicle, waited 48 hours, and then measured RNA-seq to measure the levels of the barcodes, and DNA-seq as a normalizing factor for the number of plasmids that were found in the EMPERA assay. We also transfected positive controls. These are sequences that are known to increase expression of regulatory, to have increased expression. And then we also did negative controls as well, which were just scrambled sequences. We then quantified activity of the EMPERA elements. We did this using either active elements, where we quantify the levels of RNA over DNA expression compared to negative controls. So if it's significantly different from negative controls, then we would say it's an EMPERA active element. We also quantify differences in activity due to stimulation and differences in activity due to allelic effects here. Okay, so that's the design of our multiplex parallel reporter assay. So first thing we wanted to do was to determine if the EMPERA shows consistency across replicates. So here you can see the replicate correlations, which are uniformly very high, ranging from 0.97 to 1. And so the RNA over DNA levels for replicate 1 and 2 are shown here, and you can see a very strong correlation. This gives us some confidence that we can conduct the assay in a repeatable manner. Also, as a positive control, we installed those commonly used promoter elements like CMV or EF1 alpha, and those show very high RNA over DNA activity, much more so than negative controls, kind of as expected. So this gives us some confidence that the assay is working as expected. Next, we wanted to evaluate those CAQTLs. So those chromatin accessibility peaks, interestingly, had pretty low levels of activity. So most of them were inactive and only about 6% of them were active. But when we did detect activity, so activity above negative controls, in those examples, we were more likely to detect allelic effects. So in all future parts of this study, we only evaluate allelic effects when we detect activity above negative controls. Okay, so what did we find overall? So here's the total number of elements that were actually detected to be active, so above negative controls. And those were conducted in both the vehicle and the stimulation condition. And we detected around 900 elements that showed activity above negative controls in either the vehicle or stimulation condition. Most of them were shared across the vehicle and stimulation condition, although some were detected only in either one of them. For each locus, so each region of the genome where there has an association to cortical structure, around 70% of those loci had an EMPRA active element. So implying that there are regulatory elements that are active at about 70% of loci that were associated with brain structure. That's only, though, around 7% of variants that were tested. And that's kind of expected because we think that most variants in the genome aren't going to have particular function within human neural progenitor cells. We also evaluated condition-dependent effects. So those effects that were only observed, so those effects that were accentuated or decreased in Wnt stimulation. And so we detected a small number of elements that had condition-dependent effects. Interestingly, for almost every region in the brain, for either thickness or surface area, we detected some EMPRA active elements. So overall, most cortical structure-associated loci contain EMPRA active elements. In order to determine downstream impacts of condition-specific EMPRA active elements, we mapped elements to the gene of interest using either location at a promoter region or chromatin interaction as measured through a technique called Hi-C. So we were able to map these EMPRA elements, even though they don't really tell us which gene is involved, using some additional information. And we mapped the elements to the genes that they affect. Interestingly, we found that those EMPRA elements that had higher activity due to Wnt stimulation also had higher expression due to Wnt stimulation. So in this way, it seems that we're identifying these Wnt-sensitive enhancer regions that are regulating Wnt-sensitive gene expression. So we're identifying elements that basically function as condition-dependent enhancers within the genome. Okay, so we also wanted to see if these EMPRA active elements overlap any kind of known genomic annotation. So one kind of annotation of interest in the EMPRA literature has been ALU elements. So ALU elements are a type of retrotransposon. These are elements that basically copy themselves and reinsert themselves into the genome, and copy themselves and reinsert themselves into the genome. And they've been found to occupy around 10% of the genome in total, so a pretty large part of the genome. They've also been hypothesized to be involved in human brain evolution because ALU elements have increased along the human lineage. So interestingly, the EMPRA active elements actually overlapped with the EMPRA active elements were enriched in ALU elements, and specifically within the left arm of the ALU elements, which contains these enhancer regions, shown here, for RNA polymerase III. So these enhancer elements in the left arm may be governing sort of the expression of these EMPRA constructs. Interestingly, also, the newer ALU elements, those that have these lower deviations from an older ALU sequence, these newer elements seem to be the most enriched for EMPRA activity. Also, those genetic variants that had allelic effects, those were enriched specifically in the left arm of the ALU elements rather than the right arm. And those genetic variants that had the biggest allelic effects in the EMPRA assay, shown here, also generally overlapped with these particular promoters called the A and B box that function as these RNA polymerase III promoters. So, it's possible that strong ALU element activity is maybe an artifact of the non-chromatinized sequences in the episomal EMPRA library that's not chromatinized. So those may normally be silenced in the endogenous genome. So to support the idea that some EMPRA active elements exhibit endogenous genome function, we used an assay that measured enhancer-promoter interactions using enhancer RNAs. And enhancer RNAs are a separate way that enhancers can loop to promoters and regulate gene expression. And we actually found that the active elements seem to be enriched in these enhancer RNAs and upstream antisense RNAs right at the promoters. So what this implies is that Emperor active alus overlap enhancer and promoter RNAs and may have some function in the endogenous genome. So to demonstrate the utility of EMPRA for fine mapping condition-dependent gene regulatory potential at these complicated loci, I present an example here, which is actually the same example that I showed previously. And in this example, we have, these effects on the brain, specifically the strongest effects for lingual and pericalcarine areas of the cortex. Again, these are complicated loci. We don't know the causal SNP at the locus or any of them with gene regulatory potential. But EMPRA helps us narrow down certain regions or certain SNPs with gene regulatory potential, some of which have condition-specific effects. So in this locus here, there's one particular SNP that has a condition-specific effect. And the stimulation of Wnt actually blunts the effect here. And interestingly, we're also able to identify from this SNP which transcription factor motif it disrupts, suggesting individual transcription factors which may be involved in the function of these particular SNPs. Okay, so overall, we identified a lot of different allelic-dependent effects in EMPRA active elements. So about 77% of loci with an EMPRA active element contained also an allelic difference, and those allelic differences were found across the entire brain. Okay, so, I presented before that we have these interesting findings where sometimes genetic variants, even though they're present in every cell in the body, have specific effects on certain brain regions, but not on other brain regions. And we really wanted to understand better, why does this happen? So here we took an example of a particular SNP where this SNP is associated, again, with cortical surface area differences, mainly in the occipital cortex here. So it turns out that this SNP showed an EMPRA allelic effects and disrupted a binding site for a transcription factor called RARG. So when we looked at the expression of RARG in the developing cortex, it had a remarkably similar pattern of expression, actually an opposite pattern of expression to the effects on cortical surface area. So we can actually relate that, we can correlate these two. So this axis shows the effects on different regions of the cortex, and this axis shows the expression of the downstream transcription factor. So it's likely that though this SNP is present in every region, it's disrupting a transcription factor. When that transcription factor is highly expressed, it has a stronger effect on the structure of the cortex. So this kind of spatial relationship in transcription factor expression and the disruption of that transcription factor by a SNP together can lead to regional specificity of brain structure effects. So overall here, EMPRA and CAQTLs can measure different aspects of gene regulation. Most cortical structure-associated loci contain elements with EMPRA activity and allelic effects, suggesting specific gene regulatory regions, SNPs, and stimulation states impacting brain structure at complicated loci. Also, the regional specificity of SNPs may be driven by both motif disruption and TF expression, the combination of the two. And EMPRA activity is largely driven by younger ALU elements, and this suggests that there's an eRNA, an enhancer RNA mechanisms regulating gene expression, and provides some evidence that retrotransposons have contributed to human cortical evolution. Okay, so for one last little vignette here, I'm going to tell you about how our lab is also studying how genetic variation is associated with cortical cytoarchitecture. So previous studies that I've just shown focus on how genetic variation affects macroscale brain structure as measured through MRI. But macroscale brain structure doesn't really give you the cellular basis underlying those differences. So for that, we need a different technique. We have to move to microscopy, where we can understand how genetic variation is associated with cortical cytoarchitecture. In these type of studies, instead of using a human system, we're going to use a mouse model system, mainly because the mouse brains are much smaller, allowing us to image the whole brain. So there's been these new techniques that have been developed called tissue clearing techniques that allow us to image an intact brain without doing physical slicing. So these are really great because they allow us to image an intact brain and understand how differences in genetics lead to differences in brain structure. So for this study, we focused on NF1 mutant mouse. So NF1 is the gene associated with neurofibromatosis type 1, which often presents with intellectual disability and autism. So we took 6 mice that were controls, 6 mice that were heterozygous for a conditional knockout in NF1, and sorry, one second, and 6 mice that were homozygous for a conditional knockout in NF1. We then put them through a tissue clearing procedure, which basically removes the lipids and allows light to pass through the sample. And we labeled with lower layer neurons and upper layer neurons, as well as a nuclear marker. We then conducted a light sheet microscopy, allowing us to image the intact samples in a very quick manner, although the samples still take around 16 hours per brain to image. So here's what nuclear brain structure looks like. So here's a 3D rendering of a hemisphere of a mouse brain. So the nose would be up here, and the back of the brain is here, and the ears would be on the side over here. And the brighter colors here indicate a higher density of nuclei. So you can see here that we can get a full brain. And if we zoom into an individual region, we're actually able to see individual nuclei across the brain. So here represents each an individual nucleus. Excuse me. And these can be visualized in both the XY dimension and the XZ dimension because we're getting full 3D structure of the brain. Okay, so these type of images are really big. They're around 1 TB per brain. And so we need analysis methods to be able to process these really large images. So we've developed some tools called Numorph that allow us to analyze these really large-scale images. After collection, then we go through image stitching and alignment. For each of the different channels, we're getting a full 3D brain. We register those brains to the Allen Brain Reference Atlas to get annotations of different regions. And then we also need to count features in the complicated images. So we have developed some tools to identify nuclei and co-localize cellular markers in these images. And then we can compare basically across regions, within each region across genotypes. So here's an example of what these brains look like. So on the right side, we have the NF1 conditional knockout, and the left side, we have the control. Different regions are highlighted here based on annotations from the atlas. And we're just focusing on the cortex, scrolling through from anterior to posterior. So the bottom layers here are the lower layer neurons that we labeled, and the upper layer neurons are labeled here. And you can notice there's a slight thickening in the cortex in the NF1 conditional knockout. And once we get to the rendering, you can see that the overall cortex is actually bigger. Now it's going to flip to the left side. The overall cortex is actually bigger too in the NF1 conditional knockout as compared to the control. Okay, so these images are big and complicated, so we need methods for quantifying the images. So one of those methods is just to count all of the different nuclei within the image. So those nuclei can be annotated using doing manual annotations, but doing manual annotations for millions of nuclei is clearly not feasible. So we developed a machine learning tool to take 50,000 nuclei that were manually annotated and predict the nuclei in the rest of the data set using probability maps and then thresholding to get centroids. We get around 8.4 million nuclei, and we get both pretty high precision and recall for these methods. So here's what it looks like after doing the segmentations. So each of the dots represents a different cell. Those cells are annotated. Those cells can also be co-localized with different labels for upper and lower layer neurons or for neither of those. And that can be done for across the entire cortex as shown here. And then just zooming into the cortex. Okay, so what did we actually find about NF1 versus control? Well, we found that there was an increased thickness in cortical thickness, specifically in posterior regions of the brain in somatosensory and visual cortex. So that increased thickness was actually accompanied by increased numbers of total cells, which are stained by TOPRO3, which is a nuclear marker. Interestingly, though, it was not driven by excitatory neurons. So excitatory neurons labeled with CTIP2 or CUX1 were actually not different between the control and the NF1 knockout. Instead, glia are really what drove it. So those cells that were CTIP2 negative and CUX1 negative. To further demonstrate that, we took markers that are specific for astrocytes or oligodendrocytes and found that those were actually increased in the NF1 conditional knockout. Okay, so thickness increases here are driven by non-neuronal glial populations. And interestingly, those type of thickness differences were actually what was also found in human populations. So this kind of gives some credence to the approach that we have here. Okay, so that's just one example. We're trying to expand our approach here by first expanding our training data set through manual annotation. And I'm going to try to get this video to play. Well, maybe it won't play. Okay, so anyways, we're expanding our data set through manual annotation where we're having very brave undergrads trace individual nuclei in large 3D data sets to expand our training. So that's one approach to expand. And then also we are, instead of just using mouse models, we're using a human organoid model to study how genetic variation or differences in diagnosis lead to differences in brain structure as measured through cortical organoids, which are human stem cell derived models of the human brain. So this is a zoom through of a cortical organoid showing especially labels of progenitor associated zones, these PAC6 associated zones, and where things are proliferating. We've used these type of tools to evaluate differences in reproducibility across sites for organoid generation. So here's just an example that we've used the tissue clearing approaches where we've labeled across 3 different sites organoids with different markers for progenitors or the tight junctions or overall the number of cells. And basically found that these measures can be somewhat reproducible across sites if we normalize for overall volume of the organoid structures. Okay, so just to wrap up here, so we've developed some tools to quantify cellular resolution, brain structural differences in 3D images of mouse brains. Using these tools, we found that the NF1 conditional knockout has similar spatial patterns of cortical thickness differences to the human, and that importantly, those increased thicknesses are driven by glial cells, which adds on to the MRI literature. Also, tissue clearing technologies allow the study of psychiatric disorder-associated genetic influences, and we plan to extend this to multiple other mouse models, as well as to human cortical organoids as an in vitro model of human brain structure. So overall, my lab tries to understand how genetic variation leads to differences in brain developments, specifically gene regulation within brain development, altering differences in cytoarchitecture, leading to differences in macroscale brain structure, and ultimately creating risk for psychiatric disorders. The hope is that if we understand these pathways, leading from genetic variation to differences in much more complicated traits, that hopefully we'll be able to develop some therapeutics to alleviate some of the symptoms of psychiatric disorders. So I want to thank you all for your attention, and I want to especially thank all the people that have contributed and completed these studies, especially our research associate, Nana Matoba, who led the EMPERA analyses, some alums from the lab, including Dan Liang, who's a postdoc at Yale, who led the CAQTLs, Oleg Krupa, who led the tissue clearing, Neil Lagoon, who led the EQTLs, and now a postdoc at the Broad Institute, and Rose Glass, also a postdoc at Harvard, who led the tissue clearing on the organoids. And also I want to thank our collaborators, Haejung Wan here at UNC and Jessica McAfee, who co-led the EMPERA analysis that I showed. So thank you very much, and I am happy to take any questions.
Oh, Jason, I've got so many questions. We're going to need another hour at least to get to all my questions. I've got so many. I mean, I was really interested in the fact that you guys are begin looking at your Wnt pathway in prenatally. And I'm curious about this because so much of development is happening before someone is ever born, and that you're looking at a window of development that's probably relatively narrow in the overall like lifespan of people. And I'm kind of curious about like in development where the brain itself is changing its shape. There's a conformational change through development. You sort of touched on that, but then some of your later graphics, you sort of show like an adult version of a parcellated brain. And I'm kind of curious if there's been any work to look at the spatial arrangement of the various regions that you're kind of targeting with respect to the genes and how they express themselves in that particular moment in in neural development. Because once the brain is fully developed, things move quite far away from each other, and I'm kind of curious if that is something that is accounted for in any of these models.
Yeah, really interesting. So I think I'm going to go to this sort of map of the human brain during development to try to talk about this. So,
Yeah.
there are some data sets available for mid-gestation fetal brains for in situ hybridization, allowing us to have at least some idea of how genes are expressed during this early developmental time period. And it would be very interesting, and I think there are a few studies that are trying to relate these to differences in the size of the brain later on in development. I think spatial transcriptomics would really be the most interesting here because it would provide really that single cell resolution and cross genome resolution. I think those studies I've only seen, I think there's a preprint from the Kriegstein lab that has some of this spatial transcriptomics from fetal development. But those are usually only done in individual slices at this point. So to have like a full 3D brain spatial transcriptomics during fetal development would be very exciting. I don't know of any study that's done that yet, but I think that that would be really exciting and would allow you to sort of relate the differences in expression early on at a single cell resolution to the differences in brain structure later on in development.
Yeah, I mean, it's kind of interesting about you think about the spatial multi-omic elements of this, and you sort of almost assume that, this, I mean, things are, I don't know, more or less, either they're different parts of the body or they're in an organ, they're in different parts of the organ, but the shape and structure of the organ itself isn't necessarily changing underneath it. Here you've got, the whole developmental process, which is kind of like led me to wonder about, you mentioned some findings with your RARG. Was that what
Yeah.
it was called? Where you were seeing those effects that were present in lingual and kind of pericalcarine areas, like around the cuneus and whatnot. I'm kind of curious if whether or not there might be some evidence of expression there or effects there that are related to congenital blindness, because that is a like known area of, around V1 and at the center of the optic field of view. I was kind of wondering if those thicknesses or changes in those thickness that are like more or less are related to something like congenital blindness. Do you know of any studies like that?
That's interesting. Yeah, I actually don't know, but there are like I showed before, there's incredible number of GWAS that exist. That is something that normally we don't check because we usually just use psychiatric and neurological disorders
Yeah.
and cognitive traits. But yeah, it would be very interesting to check that, and that is something that's definitely very straightforward to go after. So yeah, that's a great idea.
I'm reminded of my colleague. I think you probably know him too, Emiliano Ricciardi. He's been a kind of mover and shaker in the human organization for human brain mapping for a number of years, and he studies congenital blindness. And so he does a lot of neuroimaging studies that look at that. And I'm kind of curious if there might be some human evidence for that. If you had the kind of some cell cultures that you could look at in his population, it might be kind of interesting. But
Oh,
anyway,
interesting idea. Yeah.
random thought. The other thing I was thinking of is what would be, you mentioned some of your methods for manipulating, putting the barcodes and whatnot on some of these things as a tracer. What role do you think something like CRISPR might have to bring to bear on this? Any
Yeah,
ideas?
CRISPR would be another great approach. So there's many types of CRISPR
Yeah.
and many types of studies that you could do with this. To to say what I think would be the most interesting type of study, there are these new versions of CRISPR called CRISPR prime that allow you to insert individual base pair changes into the genome. So it's sort of a cut and paste, allowing you to insert individual base pair changes. Um, if you could multiplex CRISPR prime at a single cell resolution level, it would allow you to insert individual variants into a particular cell's genome and then determine the outcomes of that individual insertion. So Those type of studies would be, to me, the best study for evaluating a genetic variance function. And the reason for that is here we're using sort of these exogenous little plasmids that are not integrating into the DNA and they're not in the right place. So they have a lot of limitations to them. Whereas doing a CRISPR screen at single cell resolution with single base pair resolution would allow you to exactly test what you want to test, which is how does me changing just one base pair impact gene regulation? So I think those techniques are sort of coming along. There's at least some ability to do individual CRISPR prime mutations. There is not really a multiplexed version of CRISPR prime that I'm aware of yet that would allow you to do that at a single cell level. So I think those type of things, though, in the future would be where these type of studies would go. Yeah.
One final question for you. You mentioned the size and scale of the data that you're collecting, in particular with regard to some of the clearing techniques that you're using in doing the imaging and the nucleus counting that you're doing. What are some of the data science challenges that you see are like presented now, and what are the things that are coming that we need to prepare for?
Oh, that's a great question. That's a big question. So, I mean, I would especially say, so to be honest, the genomics assays are not our real problem anymore with like in terms of like overall size of analysis. The problem with overall size of analysis are these type of imaging, these type of data
Yeah.
sets. These data sets are so huge, they're around 1 TB, so you can't to even load them in to large scale to do the types of analyses, require chunking of data where then you have boundary problems and it's very, very difficult. Sharing of data becomes very difficult because, how do you share? Like if here we have 6 × 3, 18 TB just in raw data to share. So, if downstream data processing is huge. So, space issues are huge, computational issues are huge. To do the processing of all of these, it's a very difficult problem. I honestly, we're struggling with it here, just to have enough storage space and enough compute to like be able to do this. Cloud computing exists, but it's very expensive, especially when you get to huge data sizes as well. So I don't have a perfect solution, and I hope that, you and a lot of these other people that are listening to talk like this may be able to come up with some new solutions for increasing sizes of data.
Jason, man, thank you so much. This was like, this is right. This is my jam. So thank you so much for help, for doing this for us. This was really thought provoking. It's just a really amazing amount of work. And so congratulations to you and your team and everybody, all your collaborators for doing this. And thank you for sharing it with us today. Really excited about it. Thanks again. Everyone, this is the last of our seminar series broadcast for this year. We will look forward to seeing you after the new year. In the meantime, happy holidays, everyone. Take care, be safe, enjoy your friends and your family over the holidays. Bye bye now.